Hello, dear colleagues. My name is Polina Moskaleva. Today we will discuss the actual issue of arterial hypertension and tension-type headache comorbidity. Both diseases are common in outpatient medicine. The research of nitric oxide system in the development of this phenotype is growing last years. The topic of my presentation is the role of single-nucleotide variants of NOS1, NOS2 and NOS3 genes in the comorbidity of arterial hypertension and tension-type headache. Arterial hypertension, we will name it AH, is the prevalent condition worldwide and is the key risk factor for non-fatal and fatal cardiovascular complications. Tension-type headache, we will name it TTH, is the most common type of primary headache and is considered to be a common everyday type of headache. The prevalence of AH and TTH phenotype is from 50% to 93%. Thus, 30% of patients with headache have a history of hypertension, and 85% of patients with hypertension complain of headache. Nitric oxide is an important autocrine and paracrine signaling molecule that plays a crucial role in cardiovascular physiology and pathology regulation. NO is an important molecule involved in the regulation of cerebral and extracerebral cranial blood flow and arterial diameters. Reduced bioavailability of NO in the endothelium is an important precursor for impaired vasodilatation and hypertension. Furthermore, NO is involved in nociceptive processing. A NO-induced biphasic response with immediate and delayed headache is typical for chronic TTH in humans. NO syntheses are expressed in three isoforms: neuronal, inducible, and endothelial, N, I, and T, and OC, respectively. The functional activity of enzymes of NO synthesis family depends on the carriage of wild, highly functional, low functional, and no functional alleles of single-nucleotide variants of NOS1, NOS2, and NOS3 genes encoding the enzyme isoforms. Dysfunction of NO synthesis leads to endothelial dysfunction. NO and NO synthesis play an important role in the pathogenesis and treatment of both AH and TTH. In particular, NO synthesis affects brain sensitivity to stress and changes in systemic and local hemodynamics. Moreover, NO synthesis change in endothelial cells of peripheral and cerebral vessels is one of the identified causes of NO-dependent vasospasm, impaired NO-dependent vasodilatation in patients with AH and TTH, as well as changes in response to antihypertensive drugs and analgesics. NO is the main mediator of endothelial dysfunction underlying the development of AH. Endothelial dysfunction is the first step in the development of atherosclerosis. It's characterized by reduced biosynthesis and decreased bioavailability of NO. Kalem et al. Identified three causes of NO-dependent vasospasm: impaired NO-dependent vasodilatation in patients with AH, reduced NO synthesis in endothelial cells due to impaired signaling transduction and/or decreased NO synthesis activity, accelerated NO degradation in vessels wall, and structural disorders in vessel wall leading to a general decrease in dilator capacity of resistant arteries. Increased NO synthesis enhances angiogenesis. Angiogenesis is impaired, then NO levels are reduced. Signs of impaired angiogenesis and, as a result, microvascular rarefaction are revealed in AH. The genetic basis of these mechanisms has also been studied. The release of NO from blood vessels, perivascular nerve endings, or from central nervous system tissues is an important molecular trigger in spontaneous headache pain. Assessment of variations in L-arginine or NO pathway activity showed the increased content of NO and significant increase in cytosolic calcium 2+ concentration in patients with TTH, peculiarly in patients with analgesic abuse compared to control individuals. It can be hypothesized that the increased NO synthesis activity on the periphery in TTH patients reflects an analogous central upregulation of NOS synthesis activity in spinal horn, autogeminal nucleus, and supraspinal structures. These structures are involved in the modulation of nociceptive input from myofunctional cranial structures, contributing to central sensitization. Objective. The aim of the report is to define the role of single-nucleotide variants of NOS1, NOS2, and NOS3 genes in the comorbidity of arterial hypertension and tension-type headache. Methods. This list demonstrates the design of our study. The research using keyword nitric oxide synthase showed the high interest to study of the NO system. The filter using keyword arterial hypertension demonstrated that publication number of the role of NOS in the development of IH is 10% of all results. However, the publication number when using keyword tension-type headache is smaller versus AH. That is rather unpredictable. There are many evidences of pathogenetic role of NO in TTH, but there are no studies of NOS's SNV's predictive role in TTH development. Results. The NOS1 gene is localized on chromosome 12. This gene is widely expressed, especially in skeletal muscle. There are biased expression of NOS1 in brain and other tissues. As I have said before, neuronal NOS plays an important role in the development of both AH and TTH. Probably it is the reason of the high comorbidity of this disease. However, there are many evidences of the NOS1 gene synthesis in AH, but there are no in TTH. For example, Levinson and others found that the carriage of T allele RS3782218 reduces the risk of development AH, and the carriage of A allele RS7314935 on the contrary increases this risk. The role of synthesis in the headache development was studied also, but only in migraine, and no associations were found in these patients. The NOS2 gene is localized on the chromosome 17. There are biased expression of NOS2 gene in more than 10 tissues. The inducible NOS function produced by the NOS2 gene is activated to combat environmental factors. As it was said before, NOS play an important role in the development of both AH and TTH. It also may be a reason of high comorbidity of this disease. However, nowadays there are many evidences of role of NOS2 gene in synthesis in AH, but there are no in TTH for our surprise. Jai and others studied the association between synthesis RS27792249 and RS22975118 of NOS2 gene and IH in Han-Chinese Hogot, and provided evidence that NOS2 is strongly associated with AH. Topcheva and others showed the presence of T allele RS37300017 had protective effect, and presence of C allele RS18000482 increases the risk of essential arterial development. NOS2 SNV's associative studies showed the relationship of this RS in haplotypes with an increased risk of primary headache, but also only on the example of migraine. The NOS3 gene is localized on the chromosome 7. The human NOS3 gene is highly polymorphic. There are biased expression of NOS3 gene in different tissues. Vascular endothelial cells produced NO, which contributes to the regulation of blood pressure and regional blood flow. Of course, NOST also played an important role in the development of both AH and TTH. It may be also the reason of high comorbidity of this disease. However, there is a similar situation. Now there are many evidences of the NOS3 gene is in AH, but there are no in TTH. Thus, Nassereddin and others showed the association of SNVRS 179983 of the NOS3 gene with AH among a sample of Moroccan patients. Gamil and others found the association between AH development and RS2070744 in Sudanese patients. Salvi and others revealed an association between AH and RS39182226 in the NOS3 gene promoter. Quite a few studies have considered the association of NOS3 gene SNVs with headache, also with migraine. Discussion. As a result of our research, we can make the following hypothesis. All three isoforms of a protein and its genes, described above, played key roles in the pathogenesis of AH and TTH phenotype. They also negatively influence the leading environmental trigger of these nosologies: stress and related neurophysiological disorders, anxiety and depression. However, the prognostic role of NOS1, NOS2 and NOS3 genes in the development of the phenotype hasn't been studied in comparison with the second most common phenotype, AH and migraine. Our analysis testifies to the importance of the planning and conducting large multi-central associative genetic research in the role of NOS1, NOS2 and NOS3 genes as predictors of the phenotype in various racial and ethnic groups. Activating NO signaling or increasing NO bioavailability are key mechanisms contributing to the positive cardiovascular effects of many drugs. Based on this, new treatment plans targeting the NO system are currently being investigated and developed. They included NO donorship and NO stimulants. The analgesic effect of NO inhibition in patients with chronic TTH is probably due to a reduction in central sensitization at the level of spinal dorsal horn to trigeminal nucleus, or both. Furthermore, NO's inhibition may become a novel principle in the future treatment of chronic headache and chronic tangent-type headache. Conclusion. The contribution of genetic and environmental factors in the development of AH and TTH phenotype has been understudied. This issue is very important for future researchers because it should help understand the mechanisms of general AH and TTH phenotype in humans. These studies will help us make new personalized approaches to the treatment of the phenotype in future. New drugs for AH and TTH can be based on the inhibition of nitric oxide production, blockade of steps in NO-cGMP pathway or NO scattering. We hope that the results of our study help understand the personalized responses to the drug-modulating NO system in patients with phenotype. Thus, we described to start a multi-central associative genetic study of AH and TTH phenotype. At the moment, the project is underway in two regions of Russia, Siberian and Northwestern Federal regions. There are research teams from two centers, Professor Voino-Yasenetsky Krasnoyarsk State Medical University and Bekhterev National Research Medical Center for Psychiatry and Neurology. Professor Nasserova Regina, Schneider Natalia and Petrova Marina lead the project. our research is ongoing, so we invite colleagues for cooperation. Thank you for your attention.